With new emerging technologies, the world today is brimming with a lot of possibilities. We are now equipped with the knowledge to make vaccines and medicines and counter diseases that can cause epidemics. It is also possible to grow crops which are disease and drought resistant. And the science which makes all this possible is called biotechnology. It is a field of applied biology consisting of various techniques which utilize organisms or their enzymes to produce useful products for human beings. As a science, biotechnology differs from microbe-mediated processes such as the making of curd and cheese, because it uses genetically modified organisms and operates on a much larger scale. Biotechnology techniques are useful in various fields such as engineering, technology, and cell and tissue culture. Many specialized processes, such as synthesizing and using a gene, developing DNA vaccines, in vitro fertilization, and developing biofuels, all fall within the purview of biotechnology. Interestingly, human beings have been applying the techniques of biotechnology unknowingly for a very long time. For example, agriculture is the oldest biotechnological system used to create new products. Agriculture gave rise to the selection of high-yielding varieties to get a better crop and the use of specific organisms for fertilizers to restore nitrogen and control pests. However, the modern biotechnology arose from two core techniques, namely genetic engineering and chemical engineering processes. Genetic engineering is a specialized branch of science which involves the use of several techniques to modify the genetic material, that is, DNA and RNA of an organism. The changed genetic material is then introduced into the host organism to change its phenotype. On the other hand, chemical engineering processes deal with the application of physical sciences, life sciences, and mathematics to the process of converting chemicals into valuable forms. Biotechnology helps in the maintenance of a sterile environment by enabling the growth of only desired microbes in large quantities during chemical processes, while making antibiotics, vaccines, and enzymes. Let us now understand some of the principles of genetic engineering that have been used for biotechnology. Techniques such as gene cloning, recombinant DNA, and gene transfer help to isolate and select only desirable genes and avoid the transfer of undesirable genes in target organisms. This helps improve traditional hybridization procedures where undesirable genes often get included. Gene cloning refers to the production of a lineage of cells which contain one kind of DNA fragment of interest derived from a population of many kinds of DNA fragments. A DNA piece will not be able to multiply in the progeny cells of the host organism if it is transferred randomly. the replication can only take place in a chromosome with a specific DNA sequence called the origin of replication, which initiates replication. Another genetic engineering technique is recombinant DNA. This is artificial DNA created from two or more sources and incorporated into a single recombinant molecule. A recombinant DNA molecule was constructed for the first time by Stanley Cohen and Herbert Boyer in 1972. Through the various experiments conducted by him, Boyer had confirmed about the useful properties of the restriction enzymes of the E. coli bacterium. The restriction enzymes helped in cutting out double-stranded or single-stranded DNA at specific locations, also known as the restriction sites. Boyer found out that the restriction enzymes could cut DNA strands in a particular fashion, which left the sticky ends on the strands. Stanley Cohen, on the other hand, had specialized in the study of small ringlets of DNA called plasmids, which float about freely in the cytoplasm of certain bacterial cells such as E. coli. These plasmids also replicate independently from the coding strand of DNA. Cohen had developed the method of removing these plasmids from the cell and reinserting them in other cells. Stanley Cohen and Herbert Boyer combined their processes, and thus the first DNA recombinant molecule was constructed. They inserted the gene that encodes for antibiotic resistance into a native plasmid of Salmonella typhimurium. They isolated the antibiotic resistance gene by cutting out a piece of DNA from a plasmid which was responsible for this genetic trait. This process of cutting out DNA at specific locations was facilitated by restriction enzymes. In this experiment, the plasmid DNA acted as vector to transfer the DNA piece attached to it. The antibiotic resistance gene was linked with the plasmid vector with the help of enzyme DNA ligase, which joined the ends of the cut DNA molecules. Therefore, it led to the creation of a new combination of circular, self-replicating DNA, also known as recombinant DNA. On transferring this DNA into E. coli, it was observed that multiple copies of the genetic material could be made by using the new host's DNA polymerase enzyme. This process of making multiple copies of the antibiotic resistance gene in E. coli was also known as cloning of the antibiotic resistance gene in E. coli. Another genetic engineering technique was gene transfer, which involves the insertion of unrelated genetic information in the form of DNA into the cells. Gene transfer is used to provide essential genes to patients in the treatment of some diseases such as type 1 diabetes and cystic fibrosis. With constant developments in its techniques, biotechnology is fast evolving and contributing to diverse fields such as healthcare, crop production, and waste management.